report analyzes whether or not animal testing is a favorable method for drug evaluation in the United States. In 2010 alone, almost 1.37 million animals were used for animal testing. However, this already large number excludes rats, mice, fish, and birds, which, as you can see from the graph, take up about 75% of the animals used for testing. In addition, 100,000 people die every year from reactions to pharmaceutical drugs that are very common for people like us. This calls into question whether or not animal testing is a favorable method for drug evaluation.
Our research led us to the conclusion that no, animal testing is not a favorable method of drug evaluation. We concluded this by analyzing three prominent perspectives, which were doctors against animal testing, animal rights activists against animal testing, and researchers in favor of animal testing.
So the first perspective that we're going to address is that of doctors against animal testing. The main reason doctors are against animal testing is because it can be an inaccurate method for evaluating drugs. As you can see in these visuals, animal testing with mice is inaccurate 47% of the time, and animal testing with rats is inaccurate 57% of the time. This is a significant percentage of the time and can cause dangerous results. The first reason that animal testing can be inaccurate is the lack of genetic variety in the lab populations. Lab animals are usually inbred, so traits may not appear in the lab population, so the drugs are not tested on organisms with those traits, which can potentially be dangerous. Also, animals in labs experience unrelieved pain and stress. This pain and stress causes hormones to be released into their bodies, which can interact with the drugs and alter the effects a drug has on the body. Then when humans who are not experiencing pain and stress take these drugs, different effects may be seen. Finally, when drugs are tested on animals, the animals are given a very high dosage to test for any possible toxic effects. Then scientists extrapolate from this data to find the correct dosage for humans, but there can be mathematical errors with this extrapolation that can lead to incorrect dosages in humans and lead to inaccuracies of animal tests. One of the major incidents where animal testing was inaccurate was with thalidomide in the 1950s and 60s. In the thalidomide disaster, thalidomide was a drug that was tested on both pregnant rats and rabbits and did not cause any birth defects, but when tested on humans, ended up causing very serious and sometimes fatal birth defects, like seen here with the boy with the underdeveloped limb.
Animal rights activists also provide a compelling argument against animal testing. Many argue that it is unlawful because, according to the Journal of Human and Experimental Toxicology, it allows for upwards of 85,000 animals to experience unrelieved pain during testing each year. This constitutes torturing animals, which is illegal in most states, but allowed only for animal testing. However, animal rights activists argue this is not a valid exception to the law. This picture was one of many that sparked controversy because it shows a monkey being tortured in a lab, which again calls into question the lawfulness of this issue. Animal rights activists also argue that more animals are used for testing than necessary because more trials are than necessary. This happens when researchers make mistakes that cause them to repeat trials. National organizations have attempted to combat this by creating a series of guidelines that should allow researchers to not make mistakes that will cause them to repeat trials. However, as you can see in this chart, only 20% of studies actually followed these guidelines in the year after they were implemented. Therefore, many studies still use more animals than necessary and torture them in an unlawful way. Finally, animal rights activists argue that there are gaps in the laws themselves. This is because rats, mice, and some other animals are not protected under the Animal Welfare Act, which is the main legal body protecting animals during experimentation. As you can see in this chart, only 5% of animals that are tested upon are actually protected by law. Even these 5% are only protected against things such as their housing and their feeding. This leaves it up to the researchers to determine how much they feel the need to torture these animals during experimentation. This unlawfulness, combined with the inaccuracies of animal testing, make it a less favorable method of drug evaluation.
Another important perspective to consider is that of researchers who are in favor of animal testing. A lot of researchers believe that animal testing is beneficial because of all the drugs that can result from this method. One example of this is from the Saudi Journal of Gastroenterology, where a study on rats was trying to figure out if thimaquinone or melatonin would be a better choice to treat intestinal hypoglycemia.
This could be really helpful because the mortality rate for intestinal lymphemia, as shown in this graph, is 60%. So a lot of patients could be helped if a beneficial drug could be found through animal testing. Another example of this is shown in this picture, where memantine is blocking the NMDA receptor, which is a protein that is involved in anxiety. A study on mice found that memantine can help with anxiety in mice, and this could potentially replace benzodiazepines, which are the most common in anxiety treatment and can have some very harmful side effects. However, the fact that these drugs were found through animal testing doesn't really mean that animal testing is the most favorable method, because other methods could have been used to find these discoveries, so it's really not the best because of all the potential dangers as well.
In vitro testing, or testing done with human and animal cells and test tubes, serves as a possible alternative to animal testing, also called in vivo testing. In vitro testing is a less expensive option as the actual tests are less expensive. In this graph from data from the International Humane Society, you can see that a chromosome aberration done with in vivo testing costs $30,000, while in vitro testing only costs $20,000. This is a significant decrease in cost. In addition, the other two results here mirror this data, and the in vitro testing shown in gray is often a lot less expensive than the in vivo testing shown in blue. In addition, when in vitro testing is implemented, the management expenses decrease as the actual expenses of housing, feeding, and taking care of the animals also decrease. Finally, more accurate data can be seen when in vitro testing is implemented. As previously mentioned, animals often have issues with data because of their stress, and when the animals are eliminated, so is the stress factor, providing more accurate data to the researchers. Finally, many people argue that in vitro testing is too uncommon to be used as the primary method for drug evaluation. However, in this graph, you can see that over the last 30 years, in vitro testing has increased significantly, and in 2013, 200,000 tests were done for in vitro testing. Because of these reasons, in vitro testing should be incorporated into laws and funded by governments and private investors to incentivize researchers to implement it more into their research.
As a result of this conclusion, there are a few implications that must be considered. First, laboratories would need to be altered to have the correct equipment and supplies to accommodate in vitro testing. Additionally, researchers would probably have to go back to school or undergo some other type of training to know how to use in vitro testing, and regulations would have to be altered to accommodate in vitro testing so that all researchers would be performing it correctly. Because of these measures, it would really need to be considered about how in vitro testing can really be incorporated.
It's important to acknowledge a couple of limitations on our research. First, we were limited by the amount of results we could find for in vitro testing. However, this is mostly due to the fact that in vitro testing is a less common method, so there are just less results out there to be found. Also, some of the examples we used were from outside of the United States. However, these were medical facts and statistics or results from studies that would have been the same no matter where in the world they were performed. So therefore, it still applies to our research question. We just chose to focus in on the United States for the legal perspective.
As a result of all these perspectives, we decided that in vitro testing is a very favorable alternative to animal testing for drug evaluation in the United States. Here's our image, we're excited, and our information, we're excited.
You're 8 minutes and 45 seconds. Okay, Lauren, what information from your team report did you guys choose to exclude from your presentation, and why?
Well, Kelly actually had information about Vioxx, which is another drug that researchers used against, or that would support the perspective that researchers are against animal testing. However, we determined that that was a little too repetitive, so we decided to exclude it and realized that the thalidomide example that she gave was sufficient in our research.
Excellent. Caroline, which parts of the team report did you find most challenging to defend, and why?
We found it challenging to defend mostly Kira's section about researchers who were in favor of animal testing because it had led to many important discoveries. However, our rebuttal for this was that these could have been discovered with other methods. The problem was there really is a lack of evidence backing that up, since the method in vitro testing is relatively new, like we said, so there haven't been many studies on whether or not those discoveries could be made with in vitro testing.
Kelly, how did your team go about developing this research question?
So we started looking at controversial issues and we picked animal testing because we were interested and there was lots of facts to support it. And then we decided whether we were going to focus on testing cosmetics or drugs on animals. We chose to focus in on drugs and then just wording it in a way that we could back it up with credible evidence and talk about possible solutions and many implications.
Great. Kira, how did your own views about this problem or issue change as a result of the evidence found by the other team members?
When I first started with this topic, I really only thought that the medical perspective would be important, but I really discovered that it's a multifaceted issue because of all the economic and legal problems that need to be thought about before you decide an answer.
Excellent. Big round of applause for our All right, we're going to do a quick transition.